Prion propagation and toxicity in vivo occur in two distinct mechanistic phases. By Malin K. Sandberg, Huda Al-Dajabi, Bernadette Sharps, Anthony R. Clark, and John Collier. Prions in mammals cause fatal neurodegenerative conditions, including Creutzfeldt-Jakob disease, scrapie, and bovine spongiform encephalopathy. Prions are associated with a prolonged, clinically silent incubation period, followed by an aggressive clinical phase. Prions consist of multi-chain forms of misfolded host-encoded cellular prion protein, PRPC. Here the authors demonstrate that prion propagation in the brain proceeds via two distinct phases. They suggest that the prions themselves are not neurotoxic, but that they catalyze the formation of such species from PRPC. Production of neurotoxic species is triggered when prion propagation saturates, leading to a switch from autocatalytic production of infectivity phase 1 to a toxic phase 2 pathway. Prion inoculum was prepared from brain homogenate and inoculated intracerebrally into large groups of mice. 4 genotypes of mice were used, with varying expression of PRPC from 0 to 800%. As a control, groups from each genotype were inoculated with uninfected mouse brain homogenate. Groups of 5 to 6 mice were killed at multiple defined time points or at the onset of clinical disease. Brains were removed and infectivity titer was deduced by comparison with the reference preparation. This graph shows the survival time in days of the mice that became infected. Arrows indicate incubation periods in days. The mice with 0% prion expression did not develop disease. This graph shows the prion titer of mice that were killed at defined time points. Arrows indicate the onset of clinical disease. Prion titers rose rapidly very early in the incubation period, then reached a plateau in all the infected genotypes. However, the duration of the plateau phase in each genotype varied. Prion replication, solid lines, is exponential until a limiting titer of prions is attained, which is independent of PRPC expression level. At this point, there is a pathway switch leading to the production of toxic species, dotted lines, at a rate dependent on PRPC concentration. Therefore, two distinct phases of prion propagation during the incubation period of prion-infected mice can be described. The rate of prion propagation in phase 1 is not proportional to the PRPC expression level. In phase 2, where there is no increase in infectivity, the rate of formation of the toxic species is directly proportional to PRPC concentration. A related paper, Prion propagation and toxicity occur in vitro with two-phase kinetics specific to strain and neuronal type, by Hankos et al. Also showed that prion propagation and toxicity occurred in two kinetic phases, a replicative phase followed by a toxic phase. Neurotoxicity peaked when abnormal PRP accumulation reached a plateau.